DNA replication begins at the origin of replication, a site where the DNA is unzipped. Origins of replication tend to be full of AT base pairs rather than GC. Replication proceeds from the end with a 5-carbon with a phosphate attached. AT bonds are double hydrogen bonds, which aren't as stable as CG bonds. From the origin, bidirectionally, the enzyme helicase unzips the DNA template at a replication fork that's shaped like a Y. It does so by breaking the hydrogen bonds between complementary bases in both directions away from the origin. RNA primer is required for DNA polymerase. It is synthesized by primase in order to begin polymerizing DNA. The primer binds to the end of a leading strand and acts as a starting point for DNA synthesis. DNA polymerase has to have an existing 3-OH to add on to, so it cannot start synthesizing a complementary strand from scratch. But RNA polymerase can start from scratch, so a new DNA strand starts with an RNA primer. DNA polymerase starts adding on to the 3-OH end of the RNA primer. Note that after synthesis, a polymerase replaces primers and replaces them with DNA fragments. DNA polymerase adds nucleotides only at the 3-OH end, so new strands grow in the 5-3 direction. One daughter strand is called the leading strand. The leading strand can be synthesized continuously and in the same direction as the DNA being unzipped. The other daughter strand is called the lagging strand. This strand has to be synthesized in short fragments called Okazaki fragments. Okazaki fragments begin with an RNA primer and are synthesized in the direction away from the fork. The Okazaki fragments are sealed by an enzyme called DNA ligase. The two newly replicated strands of DNA can then come together and make a copy of DNA.
Despite being a mostly accurate and detailed process, DNA replication is far from perfect. Errors can occur during replication during any one of the phases and vary in severity and problem causing. Wobble. It causes the base pairs, when they're being copied, to change their orientation just slightly so that a G may not bind with a C but instead binds with an A. This can obviously be an issue because if DNA polymerase copied the wrong base pair, we start getting mutations. DNA looping is an error in DNA replication in which DNA is separated into two individual strands. On either strand, it is possible for the nucleotide bases to bend ever so slightly, just close enough that they could possibly bind with an opposing base pair on the same strand. This would cause it to fold in on itself as the different base pairs started to link up with one another, sort of like a zipper, and this base pair linkage causes a loop to form in the spaces that do not link up. This DNA looping keeps DNA polymerase from coming through and copying the proper gene sequence.